Okay, thank you, Kavita. Okay, so let me just remind you of what we talked about in my first two lectures, which was first, generally, broadly, the evolution of genetic systems, and in the context of thinking about evolvability, very broadly. But what we concluded was that even if we wanted to think about evolvability as a consequence of sorting among groups, selective sorting among groups, we were still left with the problem of how those groups themselves evolve differences in features that affect evolvability, an example of such difference being mutation rates. And so then we plunged into some experimental work on mutation rates, and I showed you results that for asexual experimental populations, genomic mutation rates can evolve to be notably higher than the wild type starting value in such an experiment. And we traced the cause of that observation to genetic hitchhiking of a mutator or allele or alleles along with beneficial mutations, despite the long-term cost of such an elevated mutation rate in terms of deleterious mutations. And so now we're going to continue on. In the second lecture, I said, well, there's this a little bit crazy theory that some friends and I came up with, that because of the delayed cost of evolving an elevated mutation rate, you might get successive waves of mutator hitchhiking that could push the mutation rate of an asexual, remember, asexual population to a very high level, even theoretically to a level causing extinction. This was such a counterintuitive and sort of evil almost hypothesis that we really wanted to follow it up experimentally, and that's what we're in the middle of right now. And so I'm going to give you some experimental follow-ups to that, some different things we've done. Sort of, remember I said we can't really test that whole theory I just referred to in one go. It's predicted to take too long. So we try to test it in parts. If I get done with this stuff, then I'll show you some results about recombination that we've started to generate. And then, on the outside chance that there's even more time, we might talk a little bit about, very generally, about how recombination itself might be favored. So where we were was with saying, well, one of the sort of divide and conquer experiments that we wanted to do was to see that if you had a population which you had perhaps yourself already set up to have one mutator allele and a high mutation rate, could you put it in a circumstance in which it evolved an even higher rate? We first did a Chow and Cox experiment, competing single and double mutators together, and in an appropriate environment, yes, the double mutator could beat the single mutator, presumably by hitchhiking. So now we take it a step further and we ask whether we can start with a strain that has a high mutation rate already and put it through some sort of selective regime where spontaneously arising double mutators are favored and spread through that strain. And so out the back end of that experiment, we observe even higher mutation rates than we started with. And here's such an experiment. This is a really simple-minded, it's a real sledgehammer of an experiment, but it worked. We have yet to publish it. So what we did was to take a strain of E. coli that is defective for mismatch repair, so it has a 100-fold elevated mutation rate. And we essentially put this strain through a gauntlet of 3 biochemically independent antibiotic selections. So first we selected it on phosphomycin. We got resistant colonies, respread those on a plate, then by replica plating selected it on rifampicin and got resistant colonies, respread those, and then selected on streptomycin. And out the back end, there were a few lucky survivors, not very many, 9 on one plate, 1 on another, when we did this in 2 replicates. And we verified that those survivors were triply resistant, antibiotic resistant. By the way, don't worry, we autoclaved all the survivors or put them in the freezer and they're safe there. So out the back end are survivors with very high, with resistance to 3 antibiotics. And now what I'm going to show you, we then fluctuation tested, measured the mutation rates of those survivors. And here are 9 from the one replicate where there were 9 survivors. Here are 9 from the other. On the y-axis, we have log of mutation rate. This is the mutation rate of the original strain with a mut-L defect, and these guys are up here about around a log higher in mutation rate. This green line down here is the rate of a wild type. So we have achieved by this experiment, bumping it up from here to here, and by what we call hard selection, basically selection that bites into the ability of the population to persist unless it has resistance. So that's some confirmation of the possibility that at least under an extreme selective regime, we expect to have perhaps higher and higher mutation rates. We went ahead and sequenced these survivors, genome sequenced them, because we just wanted to track it down to what the mutation was. And this produced a curious and interesting result, and it's too bad Rainey isn't sitting up there, because he'll jump all over this when he sees it. Of course, all the sequenced clones retained the mut-L mutation. They also, we sequenced one clone from one experiment and one from the other. They also now contain a mutation in a locus called mut-T. And mut-T is a biochemically independent mutator locus that potentially, when mutated, you've seen it in an earlier experiment I showed you, raises the mutation rate. And they all have resistance for all three selective, they have mutations that confer resistance, confirming that there's no contamination. So then we asked, well, do these mut-T mutations, which you're going to see are the same mutation in both independent experiments, do these actually account for the elevated mutation rate? And they do. When we put plasmids containing wild-type mut-T genes into the strains, we rescue the mutation rate back down to the rate that we had before we did the experiment. So that confirms that we have indeed tracked down the mutations responsible for these elevated rates. And so this just shows you that we then went in knowing what the mut-T mutation was in the one clone that we sequenced of the 9 in this one experiment. We could just PCR up mut-T in the other 8 clones, and sure enough, and no big surprise, they all had that same mut-T mutation. They were probably all descendants of one lucky guy early in the experiment that got the mut-T. Okay, so here's the interesting thing about that mut-T mutation. Here's a stretch of sequence of mut-T, and here's where the mutation is. It's a 1 base pair insertion into a run of cytosines, a CG pair. It's a 1 base insertion into that run. And it's the same mutation in both independent replicates of the experiment. In both cases, there's been a 1 base pair insertion into that run of cytosines. It's not contamination. The experiments were run at different times and so forth. So why this? Why do we see this? Not only that, we see it in a different experiment done in Patricia Foster's lab. She did a mutation accumulation experiment. In one of the lines, they picked up a mut-T mutator, and it had an insertion in this same run of Cs. And in one of Rich Lenski's long-term lines, they picked up the same mutation, the same repeat region of, so this is a different thing here, but the same, sorry, I bumped over something. There we go. This is the Foster lab. That's the Lenski lab, sorry. In both the Lenski lab and the Foster lab, a mut-T mutator, which was the result of an insertion in this run of Cs, was discovered under 2 different circumstances. So we ask ourselves, why does this happen? And this is where, if Rainey were up there, I'm sure he'd be interested in this. So this is convergent evolution in different experiments of the same mutator sequence in mut-T. Why does it occur? Well, as Paul talked about last night, frameshift mutations are more frequent in repeated regions in DNA. And so here we have a run of Cs, and so a possible explanation is polymerase slippage at a mechanistic level. But just at a descriptive level, we know that this is a mechanism for increasing the mutation rate at a given place in the genome, a run of repeats. And so that raises the question, these mut-T things are obviously mutable mutators. And remember, in the stuff I showed in an earlier talk, the mut-L's mutators that we found in Lenski's populations also resulted from shifts in a 3-unit repeat. In one case, it went to 4, in the other to 2. So are these evolvability adaptations? I'm sort of skeptical, but I can't rule it out. Let me give you the kind of argument that would that would be made in support of this. I have to think a second to get it straight. Under tough times, it might be, quote, beneficial to evolve a high mutation rate, because then you could acquire necessary beneficial mutations more rapidly. But it might be also beneficial to have a higher rate at which a high mutation rate arises. And so that would mean having a mutable site in a gene that controls the mutation rate. And so they call this evolving a high mutation rate under tough times second-order selection. Some people call it that in the literature. Having a mutable mutation rate locus in some sense would be third-order selection. It's 2 levels removed from the actual selection that's going on in the environment. I can't rule it out. It's interesting. I'm gonna leave it there. Okay, so the next thing we did in this stage of trying to test out these ideas was the following. We thought, well, okay, the sledgehammer experiment with antibiotics, that's it's sort of really obvious that it should have worked. Let's instead do, oh yes.
Maybe because I see you're going somewhere else. How is this second-order mutation different from a mutation in a genetic network, which is much more important for the network? You know, maybe it's setting the metabolic rate of the bacteria.
It is different in the sense that we are assuming here that the mutator locus itself has no direct effect on fitness. All it does is change the mutation rate. So it changes the probability that the genome in which it resides will acquire mutations, both good and bad. So its change in frequency will have to occur as a result of linkage disequilibrium, technically, with good and bad mutations. So I think it is different. It requires a population genetic explanation if you make the assumption of neutrality, whereas what you're describing is mechanistically, it's an individual level advantage. If you make a mutation in a transcription factor that changes the behavior of a network that therefore sets up the bearer of that mutation to react differently to the environment, sort of like what Paul was talking about last night, that's an individual level advantage. Okay, so in this one, we did this experiment. We decided to do what we took to calling the re-Lenski experiment. That is, doing the Lenski experiment over. We didn't follow exactly what Lenski did, but what we did was, here was our thinking. If you'll recall from what I described before, in the Lenski experiment, he started with things with low mutation rates, and I and others discovered that high mutation rates evolved during the course of that experiment, and they evolved by all that stuff we talked about before. Well, we thought, why don't we take the ancestral strain of the Lenski experiment and engineer it so that it starts with a high mutation rate and then evolve it under pretty much the same circumstances that Richard Lenski did and ask if it evolves an even higher mutation rate spontaneously as a result of new mutators arising by mutation and being favored by selection. So this is what my student Mitra Egbal did. She set up 30 populations. They had a single mutation, a mutation in mut-S. That's in the mismatch repair pathway. And they were all isogenic, except she had ERA- and ERA+ ones. And she grew them in a liquid minimal growth environment. She grew them in plates instead of flasks with a bit more sugar than what Lenski did. And she propagated them by batch transfer and, of course, froze them down once in a while so that we could analyze it. And then she went in and fluctuation tested. She did a lot of fluctuation tests. She sampled 150 clones after 900 generations from the 30 different populations, 5 clones at random per population, and she measured mutation rates of all of those to a certain drug resistance using fluctuation tests. And here's an overview of the data, and then I'll show you a graph of the data. 9% of the clones she measured seemed to have higher mutation rates than the mutator ancestor. 23 seemed to have lower mutation rates than the mutator ancestor. 68% had similar mutation rates to the mutator ancestor. And when I say higher, lower, and similar, this is based on 95% confidence limits that come out of the way of analyzing the fluctuation test. And so here is a graph of the data for one series. These are the ones that could not eat arabinose, which is irrelevant in the glucose-limited medium, but we use it for fitness testing. And a thing that has always concerned me about these data is that you have lots and lots of tests. And so you can get spurious appearances of significance when you have this many tests. Nonetheless, there's the suggestion of variability here. The other series was much more interesting. So these are the ERA plus clones. And in here, you can see that one of the populations, all 5 clones sampled, had significantly higher mutation rates than the mutator ancestor. And in another population, all 5 clones sampled had significantly lower mutation rates than the mutator ancestor. So this part of it had me jumping up and down, and this part of it had me scratching my head. So I was jumping up and down and scratching my head at the same time when we got these results. So what can we make of these measurements? Clearly, there's variability across these replicate populations. How much variability I'm cautious of just because the multiple testing problem. And indeed, within populations, I don't make too much of the polymorphism. I've had some arguments with Mitra about this. I think she put in so much work that she believes in it. But there are these 2 populations where one appears every clone seems to have a strongly elevated mutation rate, and in another independent population, every clone seems to have a strongly lowered mutation rate. By the way, here's one thing we did to try to reassure ourselves about the reliability of the fluctuation test. She took a set of clones and remeasured their mutation rates in independent tests. And so those are given by the 2 different colors here, the red diamond and the blue square. And by and large, on remeasuring the test, it's a very reliable test. And so that's reassuring, but she didn't do that for all of them. That would have been a lot of work. Okay, so how do we explain these data? I put her up to doing this experiment because I wanted to see if an even higher mutation rate would evolve. Well, in one case, that seems to be what happened. We have yet to delve very deeply into that population and look at the dynamics of fitness and the mutator allele, which, by the way, we haven't identified yet. But the only real explanation I can think of for that one, because this is a large population and it starts with a not astronomical mutation rate, is likely to be mutator hitchhiking, which we have looked at before. But what about the decreased mutation rates in that other population, the scratching your head part of the thing? Well, so clearly what you would call an anti-mutator allele in this context, something that works against the current effect of raising the mutation rate, has spread in that population. And we think there are 2 ways that that could have happened. One is, while we assume, and as I said in response to that question, theoretically we often assume that alleles that affect the mutation rate themselves are neutral. They have no direct effect on fitness. In reality, that's an empirical question. And given the amount of pleiotropy in genomes, it's entirely possible that a locus that's involved in controlling the mutation rate is also pleiotropically involved in something that directly affects reproduction and survival of the cell, rather than merely changing mutations. So that's one possibility. The other possibility is by virtue of decreasing the deleterious mutational load. Of these 2 possible explanations for the lower rates, I favor the first one, and I'll tell you why I don't favor the second one. And that's because these populations are not well adapted. Remember, they're sort of at the beginning of Lenski world. They're being put into this glucose-limited minimal medium for the first time. And in fact, I'll show you fitness data that show that all of these populations had very strong gains in fitness over the 900 generations of this experiment. What that means is that I doubt that the reduction of genetic load due to reducing the mutation rate a little bit was sufficient to overpower those other strong gains in fitness. Sorry, Anand. No, there is no. Every line gained in fitness. Every one. The ones that reduced, and actually, I'll show you in a sec. You're onto it. I'll show you. There's no pattern. There appears to be no pattern between fitness and mutation rate. And we tested that statistically. So the line that apparently substituted a higher mutation rate doesn't seem to have gained fitness faster. The one that decreased doesn't seem, yeah. The question was, are our results comparable with Lenski's results? Roughly speaking, there are some differences. He grew his populations in flasks, which are better aerated than these plate cultures that we've used. The plate cultures are infinitely more convenient. And we used more sugar because we had a smaller volume of medium. So those are, so I wouldn't stake too much on exact comparisons, but the sort of, to a rough approximation, the amount of fitness gain over the first 1,000 generations is quite similar. Yeah, but the thing you have to remember is that he started with a wild type and we started with a mutator. Again, so that's another difference. So, yeah. Okay. Yeah. Yeah, so the question is, does this connect with bed hedging, as Paul discussed? Yeah, I think the problem here is that, I would suspect maybe bet hedging, there's a timescale problem, as you say. I would suspect bet hedging if we had sort of mutable mutators involved, maybe. And so maybe when we go back to those sequences that appear to confer mutability on the mutator locus itself or on the mutation rate locus itself, that's perhaps consistent with the story. The reason I'm skeptical of it is this third order selection argument I made for you. You have to be not just selecting on the ability to produce a beneficial mutation, but selecting on the ability to have the ability to more rapidly. See what I mean? So, yeah, I don't rule it out, but it puts my head in a spin to think about it, I have to say. Yeah.
When you talk about a mutated allele, how does it exactly, what does it exactly do?
What do they do? Right, so mismatch repair, which is the one we started with, that's a pathway that, as I described in one of my earlier lectures, scans the genome for misincorporations. So if you have a G opposite an A, for example, that produces a kink in the DNA. The pathway recognizes that, and it goes on from there to take off the newly synthesized strain, recognizing it by its unmethylated state and so forth. Mut-T is involved in basically preventing a certain kind of oxidative damage that leads to base substitution at G sites. So I can tell you more about it later, but their pathways are well known, and these two pathways are independent of one another, Mut-T and Mut-S. I mean, an interesting question might be, if they were related in some way, could that explain this occurrence? Now, let's go on here. So you guys have already asked about this, so we looked at whether they had gained in fitness. So we did fitness assays, not in flasks, but on the plates. We competed them against each other, and we used the pink-red distinction to do the assays. And here's a plot. So this is the rank order of fitness relative to the ancestor among the 30 populations. And Mitra, this is Mitra's figure, but blue means that the mutation rate of the isolated clone is putatively lower. Red means that the mutation rate of the clone is higher. Yellow means the mutation rate is similar to the ancestor. Populations appear to be heterogeneous for mutation rate, have a colored shadow behind them. Take a look at that, there's really no relationship there. And in fact, you can analyze that statistically. So here's log of mutation rate versus the selection coefficient, which is the fitness advantage of the evolved strain minus 1. And there's no relationship there, none at all. Now here's something interesting. So we thought, well, okay, so we have mutation rates for the sequenced clones coming out of this experiment. Let's ask whether there's any correlation between the fold change in mutation rate that we estimated by the fluctuation test and the number of substitutions or mutations that we detected by sequencing the clone. And there is none whatsoever. you might think that the thing with the higher mutation rate would have more accumulated mutations, and in fact it should, right? But if you think about this, it could be, we don't know when the mutation rates changed in these populations. If the evolved change in mutation rate was, say, in the last if it was completed in the last 100 generations or something, then there might not have been enough time to actually accumulate a difference. If we were to continue this experiment on, we'd have every reason to expect that just neutral mutations would accumulate at a higher rate in the strains with higher mutation rates. We can do it, yeah. We haven't yet. In principle, we could go back through the experiment and follow the rise in the mutator over time, and we could figure out, when it first is detectable. This is partly a question of money because we have to throw a bunch of population sequencing at it. But the big thing, the real reason we haven't done it is we don't know which is the new mutator or the new anti-mutator. We've got some candidates, but we haven't been able to pin down what caused it yet. Yeah. Can you run that by me again? The starting fitness
versus the Lenski's at the end of Lenski's experiments.
His ancestor. I think that's not a valid comparison because although we call this the Lenski experiment, as I said before, the environments aren't exactly the same. It's a different growth vessel. It's a different amount of medium. There's more sugar. If there's anything I've learned doing experimental evolution, it's that very subtle differences in conditions can actually result in sort of invalid comparisons between things. So yeah, we've been unable to test that. Okay, so we sequenced the genomes of 20 of the evolved clones, sort of not at random, but selected to present a variety of mutation rates and populations. We do detect the ancestral mut-S minus mutation in all of the sequenced clones. So that would be your first guess that there might be a reversion of that to reduce the mutation rate or some compensatory mutations in it. Couldn't find any. We found a number of candidates. So this is where, at least for me, I start sort of going, oh God, this is going to take a while. Because what we did is to look at Go terms, right, gene ontology terms, for known loci in E. coli. And E. coli's mutation rate genome is probably the best known mutation rate genome of any creature on Earth. And so we found mutations that are, likely candidates, and we're in the business right now of sort of swapping these in and out of the evolved strains to see whether we can nail down, a causal basis, a genetic causal basis for what happened. We haven't done a lot of those experiments. Okay, so what do we conclude from this? Well, as I said, what I conclude is best characterized by the image of me jumping up and down and scratching my head at the same time. Because the big idea that you might have this up and up phenomenon, it's partly supported by this experiment. There's one line that did that, and I still think we could probably make something of that in a paper if we can nail down the genetic basis. But there's this downward evolution in one population and perhaps in other clones. There's a hint of an explanation for that, and I won't go into the details, but in retrospect, I wish we had started with mut-L rather than mut-S as the ancestral mutator, because there are experiments already out there, just in the last 3-4 years, suggesting that wild-type mut-S has some sort of direct advantage, or compensators of mut-S minus have some sort of direct advantage over mut-S minus. And so it may be that we're really, in the case where the mutation rate goes down, looking at an instance of a direct advantage of compensating some sort of effect of mut-S minus. Okay, onward. So next approach. So this is an experiment by my former student, Tanya Singh, who's getting married starting tomorrow for the next 3 days up in Delhi. Tanya came to me and she said, I'd like to do a mutation accumulation experiment with our lab's double mutator strain. Now remember, this is the strain that has a defect in mismatch repair and a defect in proofreading. And a mutation accumulation experiment is an experiment where you transfer a population from interval to interval, trying to maintain a very small effective population size. And what that does is to essentially make natural selection unable to see most mutations affecting fitness. Only ones that are bigger than about the reciprocal of the effective population size will be seen. And so the effective population size in her experiment was around 20, maybe a little smaller. And so what she did from day to day, she took the double mutator strain, she grew it on two types of medium, one sort of rich, good times medium, LB, if any of you have done microbiology, and the other a minimal glucose medium, a lot like the one I've been talking about in the other experiments. So she started with a plate with some colonies, and then she at random picked one colony and did this thing where you streak over it multiple times till you dilute out on the surface of the plate the cells till they're singletons at given locations on the plate, and then those will grow colonies. And then the next day she picked at random one of those, streaked it again, and so forth, so on and so forth. She and her undergraduate student who was helping her, okay? And things were isolated, frozen, all the usual kind of thing. And I can just summarize the results here. As expected, you expect fitness to decline in an experiment like this. In fact, remember I asserted in an earlier lecture that most mutations that affect fitness are harmful. These kinds of experiments are in essence another way of testing that hypothesis, because if mutations that affect fitness are coming in indiscriminately, not being seen by selection, then if most of them were beneficial, you'd expect fitness to go up in a mutation accumulation experiment. Instead, it almost inevitably goes down. And that's what happened here. So fitness declined. I'll show you some data. 2 populations went extinct among the LB lines, 9 went extinct among the MG lines. What does going extinct mean? So before we did the experiment, we said we have to have a definition of extinction. And the definition is the following. You have a plate and you've picked a colony at random, and because it's at random, you might be picking a very small colony. You have to pick whatever colony the algorithm tells you to pick. And then you streak it and nobody grows. And that's extinction. And to verify that it's extinction, you try that again. You repropagate the line from a few days before, you pick at random, and you see if pretty much inevitably around that time point nobody grows. And so that's what we did. All right, so here are just growth rate measures, proxies for fitness, and you can see a general downward trend. And a typical thing that happens in these experiments is that you start with a fairly small amount of variability. Each of these is different lines, and this is maximum growth rate. This is time here, the bottleneck number. Typically in a mutation accumulation experiment, there's not much variation among the lines at the start. Here you see there, right at the start, at bottleneck 10, there's already a ton of variation. We think that's because the mutation rate is so extraordinarily high. This thing has a mutation rate that's 3 orders of magnitude higher than wild type, and so perhaps that explains that. But anyway, these relationships are significant. I forgot to put the stats on there. Okay, here's the interesting thing to me. So let me tell you what each of these 4 panels are. In each panel, the y-axis is log base 10 of the mutation rate. We're assaying 2 different mutation rates. Mutation rate to nalidixic acid resistance, it's an antibiotic, and mutation rate to streptomycin. In the 2 different environments, LB and MG, okay? And the ancestral rate in each panel is given by the solid horizontal line, and the 95% confidence limits on an estimate of that rate are given by the dashed lines above it, okay? And then each point is the mutation rate of the population at the end of the experiment. So you can see that there is not just evolution of fitness in this experiment, there's evolution of mutation rates. And now go down here. The black markers are those populations that survived to the end of the experiment. The red markers are those populations that went extinct. Now, how do you assay the mutation rate of an extinct population? You don't. But you can go back to an interval as close as possible to the time of extinction and assay the mutation rate there, and that's what we did. And a remarkable pattern emerges, and it's shown best in these bottom two panels here. And that is to say that those populations that went extinct had, by and large, evolved higher mutation rates. And those populations that persisted had, by and large, and the relationship is statistically significant, had, by and large, evolved lower mutation rates. So this was pretty interesting to me. So obviously we expected fitness to decline. Extinction is no big deal. That's been seen in this kind of experiment too. Evolution of higher mutation rates has certainly been observed in mutation accumulation experiments, but not the evolution of lower mutation rates. So that was pretty fun to see. So now, what about this observed relationship between population persistence and the direction of mutation rate evolution? That is wholly novel in this kind of experiment. And so in the case of the populations that evolved higher mutation rates, how did they do that? Do we invoke hitchhiking here? I think we could, but I think a better explanation is just pure mutation pressure. And this takes a little explaining. And I'm not ruling out hitchhiking, but I can't, these populations are at very small size, their fitnesses are going down, so it's kind of hard to imagine the substitution of beneficial mutations driving the mutation rate higher. I'd be happy if that were the case. Here's why I think it might be mutation pressure. You have such a high mutation rate in these populations that essentially, by mutation alone, you could have a sort of feedback loop, because the very loci that control the mutation rate are themselves subject to mutation. And if mutations are on average harmful to the functions of those loci, then a very high mutation rate would be expected to, just as a result of mutation pressure alone, produce an even higher rate. I wish I could think of an experimental way or some other way, perhaps we can talk here, to sort of discriminate between those possibilities. Right now, I don't have any clear idea how. What about the populations that persisted and had evolved lower rates? Here, my thinking is just at the level of a colorful analogy, but if the relationship between number of mutations, deleterious mutations as they accumulate in this experiment, and fitness has log fitness here, if it's faster than log linear, or just thinking more simply, if there's some maximum load of those mutations you can get before you're pretty certain to go extinct, then you can imagine populations being pushed close to that maximum, and this should really, really put a lot of pressure to select for those individuals in the population that have a lower mutation rate. In essence, it's a sort of cliff argument. The populations are pushed to the edge of the cliff. Those who get lucky and have a lower rate are taking a few steps back from the edge of the cliff. I would predict if we take the surviving populations and propagate them some more, they'll go back to the cliff and eventually go over, perhaps with ultimately evolving higher mutation rates.
Would I see a non-monotonic change in fitness, or?
Well, you saw our fitness data. I don't know if we can resolve it. Yeah, the question is, would the change in fitness be non-monotonic? I mean, overall, it appears to be a steady downward trend. But the data are really sloppy. I mean, there are newer methods of fitness, getting fitness measurements that we and others have developed that perhaps could be used in an experiment like this high throughput stuff. We can't use it on these strains because they're not set up to. Of the cliff, I don't know. That's what I was saying. I'm not sure. I'm not sure. It relates back to the old idea of truncation selection and synergistic epistasis and yeah.
I think you proposed about the idea about.
I'm sorry, I missed the beginning.
Yeah, you proposed initially about evolvability like a switch. So this is essentially the same kind of idea. So if you started with a double mutant and you see the evolution of the mutation rate to be lower.
Mm-hmm.
So is it lower by the same, your double mutants, the same double mutants are being affected, or it's something else altogether?
We don't know. So I haven't gotten to that yet, but we've actually sequenced these lines as well. And we couldn't find any smoking guns. We have a number of candidates. Some of them are similar to the ones in the previous experiment, and both students have now finished their PhDs and gone on. And so at some point, I've got to get somebody interested in doing this analysis. But at this point, the analysis becomes quite tedious because what you've got to do is to move mutations in and out and measure mutation rates and hope that you can pick up the effect. So we don't know, is the short answer. I didn't catch the beginning on Antu.
Synergistic episode.
Yes, that's what I'm proposing.
Okay. So I just want to know, has it been observed before in asexual populations in such experiments?
Well, so I can't remember. Kavita, do you know, are there any mutation accumulation experiments where the decline has been shown to be significantly faster than log linear? I don't know. I should go back and look. But the problem with that is that it doesn't necessarily mean it would be true here because it's a different organism, a different environment. That's a good point. Should we move on?
Let's have a follow.
Oh, I guess we don't. Yeah. Sorry.
Thanks. So in answer to Kavita's question, you said you and others have developed newer ways of measuring fitness experimentally. What if I want to look up some of those?
Oh yeah, just briefly, this is what we've used here is growth rate measurements. Those are actually not the best. The gold standard is some sort of competitive assay where you compete the evolved strain against the ancestor. And typically Lenski did that by doing colonies that would show up as pink or red, but you can only look at perhaps 300 at a time practically there. What people are doing now is using fluorescently marked competitors and running them through a cell sorter. And then you can look at 10,000. And you can also do it at high throughput with 96 well plates. So you can replicate at 96. We have a gizmo in our lab now where you can have 96 replicates of the same fitness assay, and you're counting 5,000 cells in each one, and the machine will go through and do them all in 1 hour. We've only had that thing for a little while, but so you can see how that will provide you with much better statistical resolution of the selection coefficient. Yeah.
I'm sorry, I think I didn't understand. Could you go to the previous slide, please? So you're here plotting the populations.
These are the populations here that went through the mutation accumulation, and then this is mutation rate. Okay, log base 10 of mutation rate. And this line is the ancestor to the experiment's mutation rate with confidence limits. And then each point is an estimate of the evolved population's mutation rate. That estimate has to be taken from one individual in the population, but because you're going through a bottleneck every 20 generations or so, it's highly representative of the population itself. Okay, let's keep going here. Okay, that's the end of this one. I'm going to switch over to lecture 3. We'll see how far we get in it. Okay, I want to switch gears and now talk about some very new work that's in revision. And it's work I've done with my former grad student Eugene Rains and with Dan Weinreich up at Brown University. Eugene's on a postdoc with Dan. And this work relates to what I've been talking about because in essence it's about trying to find conditions under which mutators in an asexual population will not be favored, And so what Eugene and Dan and a guy named Scott Wiley came up with was this notion that perhaps the sign of indirect selection on mutators might switch with population size. And the hypothesis is that perhaps in small populations, mutators will not be favored. It turns out that it works pretty well, and the explanation, while it's couched in mathematical terms, there's a pretty good intuitive explanation. So let me launch into this. This is just development here. So this is just what you expect for mutations under direct selection. That is to say, here's population size, here's the regime in which disfavored and favored, or this is disfavored and favored. And as you decrease population size, the tendency for either a beneficial or a deleterious mutation to be gripped and changed in frequency by selection declines. And in the case of a haploid population, when it declines to about 1 over twice the selection coefficient for a haploid population, then essentially that's the line where drift starts to take over. And you go to really small population sizes below that, and genetic drift prevails in the fate of the mutation. This is for a mutation that directly affects fitness, growth rate, survival, reproduction, something like that. So now here's the hypothesis for indirect selection. And I haven't used the term indirect selection, but indirect selection is what we've been talking about with mutators when we talk about their frequency or things that affect recombination rates, frequency for that matter, being affected by association with beneficial or deleterious mutations elsewhere in the genome. And so the hypothesis is this. Here again is population size, and we're talking about the fate of a mutator in a population, and we hypothesize that as population size goes down, there'll come a point where the mutator will actually be disfavored. And then going down to even smaller population size, we'll get to the point where the population is so small that the mutator is in essence neutral then when we get to that. And so, yes?
I have a question if you go back. It's an elegant thing, but then I was wondering, you could probably only go from large population sizes to small. You can't trace the other way around, like from small to large. If you try to grow, it doesn't work that way, right?
Yeah, so that's true, but what this figure is really about is what we expect at a given population size, the ability of a mutator to be sort of gripped by selection and changed. And so what I'm going to show you in the end are experimental results at very large, intermediate, and small populations in this range here. And we'll see if they bear out the idea. Yeah. So we're going to use as our measure of whether a mutator is favored or not, the fixation probability normalized by the neutral expectation. Okay, we'll call that NP fix. And so the neutral expectation for fixation of an allele is 1 over n, if it's neutral, in a haploid population. P fix is the fixation probability, so we multiply those together. For a neutral allele, then NP fix will be 1, and so we show that here. 10 to the 0 is 1. This is a neutral allele. Truly neutral allele doesn't matter at any population size. NP fix is equal to 1. Okay? And so we'll say it's favored by selection if it turns out to have a fixation probability above that, and disfavored if it's below. And we will measure that by actually doing experiments and asking how many times the mutator won, how many times it lost out of a large number of replicates. And so here's the relationship between direct selection and N, which some of you know. This is a relationship that was derived by Kimura. That expression gives it, and it's simply mapped onto this graph. NP fix here, here's population size for various values of S, the selection coefficient. Okay? And that directly relates to this earlier figure I showed you. And here's what we propose for indirect selection. And notice that in the previous figure, there's no reversal of sign of selection on the allele. It doesn't go, in the case of a beneficial allele, to being favored, from being favored to being disfavored as population size goes down. It just gets less and less and less gripped by selection until finally by this NP fix criterion, until finally it's neutral. Okay, so I'll show you a little bit of the theory that's behind it, some simulation results, and then some experimental results. So here's a model. Imagine that the mutator is fixing by hitchhiking, a familiar scenario to us after what we've been talking about. So the mutator's got to produce a beneficial mutation, and we'll approximate the probability of that as this, because beneficial mutations are rare, so they essentially drop out of the bottom of that. And then it has to survive drift and sweep to fixation. And so we can put both of these things together and we get an expression here, P hitchhiking fix. And we map that onto this thing here. Okay, so for population size and P fix. And then we have to think of the possibility that the mutator fixes by random genetic drift instead. And so we have P drift fix. Well, drift has to overpower any indirect selection against deleterious mutations. And that's given by the difference in the deleterious mutation rate between the mutator and the non-mutator. That's minus delta U del. Okay? And so we put those together, and that's mapped as this red line. Okay? For those values. And so now we put them together and we have P mutator fix. It equals P drift fix plus P hitchhiking fix. All right? And we get this interesting relationship here. The relationship is that the selection on mutators changes sign as n declines. So notice that P drift fix is always below the neutral threshold, and that's due to selection against deleterious mutations. But up here, there's a region in which the mutator is not favored by selection, and past a certain n crit here, which is U del over P direct fix times U ben, the mutator becomes favored. Why is that? P hitchhiking fix is depressed by the requirement to first generate a beneficial mutation. And going back to fundamentals, remember that beneficial mutations are much rarer than deleterious mutations. So if I decrease the number of mutators to a low enough level, they will be very unlikely to get a beneficial mutation, but still quite likely to get a deleterious mutation. And so at small size, they pay the price, but they don't get the benefit of being a mutator. It's really, I think, that simple. Okay? So we can go through this. P drift fix is generally below the neutral threshold. P hitchhiking fix is depressed by the requirement to generate a beneficial mutation. Once P hitchhiking fix drops below the neutral threshold, then selection against deleterious mutations keeps P mutator fix here below the neutral threshold until the cost of load is overpowered by drift at very small population sizes. Ultimately, as you go to a population size of 1, nothing matters in selective terms anymore. And that's why that curve takes that shape. So it's borne out in Wright-Fisher simulations with the following values. The dots are the results of simulation. And we do indeed see sign inversion. If this is our line determining for us whether the mutator is favored or disfavored, after we cross that n crit value, the mutator becomes disfavored, whereas above that value, it's favored. Okay? And here are population sizes here. So obviously the population size that you need for that n crit depends on the parameters that go into the n crit expression. And so I got some slides from Eugene. He did Kavita. It's kind of the reverse. So the question was about the drift barrier hypothesis. I think of the drift barrier hypothesis as about, something that would change the mutation rate being affected by selection or not at small size. And clearly, the drift barrier, it's valid. It must be going on as we get down here, right? I mean, we get to the point where this thing doesn't matter anymore, whether it's a mutator or not. But up here, there's this interesting sign, and we should talk about it some more, because Dan and Eugene and I have talked about this a lot. Yeah. Okay, so these experiments were done in yeast instead of grainy. Yeah, so the experiments were done by batch transfer,
Right.
but we were very careful to calculate the effective population size under the circumstances of batch transfer.
Right.
And there were a number of ways to do that. And so those are the population sizes that we actually assume
Yeah.
are relevant in the experiments you'll see. So it's controlled for the transfer, but it's a standard batch transfer protocol like Lenski and so forth.
So what if you were to consider the possibility that passing through that bottleneck involved selection?
Yeah, certainly there's theory about that. What I'll say is that the simulations were pretty good, and then the results matched the simulations pretty well. So I'm not sure how important that would be.
So what I'm thinking in particular is where, in order to be a member of that small population, you have to have a certain fitness.
Yeah, or inhabit a certain zone in the culture vessel, or sure. I mean, there are all sorts of subtleties.
And that doesn't make a difference.
We haven't looked at it in that way.
Okay.
I'm not sure how that would relate to mutation rate, though, is the thing.
Well, it may mean that the load is not so significant. Because in order to be...
explain the sign reversal as you go to...
Well, that's what I'm wondering about, the connection between, if one were to introduce some ecology in there, imagining, for example, a mutator that has to go through a bottleneck of one. But there are two ways, you could just randomly sample one from a population versus some selective, if you like, bottleneck event where getting through that bottleneck requires the cell that gets through to have a fitness of,
Yeah.
Well, we can talk when we cycle.
It could be. On the face of it, I can't see how that explains the results, but it could make them more interesting to think about it.
Yeah.
Let's press on. So it's yeast. We also work with yeast in my lab. All the usual stuff about doing experiments. These were done on plates, or on microwell plates, and shaken in an incubator. So what we did was to conduct competitions between a 100-fold mutator and non-mutator strains of yeast. It was mismatch repair in yeast. And the competitions were initiated at approximately equal frequencies, so 50% of mutator, 50% non-mutator, because we're looking at NP fixed. So the neutral expectation is 50%, right? And they're propagated by regular transfers, and as Paul and I were just discussing, N is controlled by the size of the transfer bottleneck. And we followed the mutator frequency with fluorescent markers. And so remember that the theory predicts that in large populations, mutators on average should be favored because they will hitchhike with beneficial mutations. And so here are the data for large populations. In these experiments, mutators win in 162 of 276 competitions within about 250 generations, and they lost in only 25. And even in the 89 unresolved competitions, the mean mutator frequency was higher. So it looks like they win at large population size. Intermediate populations. Well, intermediate populations, and by the way, we calculated the population sizes based on some estimates of the relevant parameters. So in intermediate populations, mutators won in 112 of 275 in the first 225 generations and lost in 124. The mean mutator frequency in the 39 unresolved competitions, that is after 225 generations, was right around 50%. Obviously not different from the starting frequency. So that one's kind of what's going on there. So then Eugene went down to smaller population sizes. And indeed, at small population sizes, the mutators are disfavored. This is the mean frequency here. Mutators win in only 53 of the 273 competitions in the first 250 generations there. So this seems to fit with the idea that in a small population, you're at risk for deleterious mutations, but you don't get the benefit of beneficial ones. So remember, here's our model. And we would say that in the large populations, we were up here in this region, and in the smalls, we were somewhere in this region. And perhaps in the intermediates, we were right around that NCRIT line. Eugene then went in and measured fitnesses. So the prediction would be that in the small populations, the non-mutators win. I mean, it's not an exclusive prediction, but you might expect the non-mutators to win in small populations by outlasting the mutators, which are lost to deleterious load. That is to say that they don't necessarily get any fitter. And so here is the fitness of the non-mutators in the small populations relative to the ancestor. Yeah, they didn't really gain much fitness. In the large populations, fitness relative to the ancestor is significantly higher, consistent with the idea of a hitchhiking event at large population size, but not small population size. So just a summary, I showed you a simple analytic model and that simulations fit it, and this independent inversion in the sign of selection that's borne out in the simulations, and it looks like it's borne out in the experiments as well. I find this interesting because it does suggest maybe a sort of general phenomenon in which things that produce variation at random, which means most of those new variants are going to be harmful, those things are more likely to be favored in those genetic mechanisms like mutation, recombination, are more likely to be favored in a large population than in a very small one, for the very sort of bias that I mentioned. And that's what's said here, is that this mechanistic bias might not be unique to mutators. Any mechanism that introduces random genetic or phenotypic variation is more likely to negatively affect fitness than improve it, and therefore at small population size, it might fall into this relationship. And so this, of course, got us thinking, and this is where we circle back to the evolution of genetic systems, because you might imagine that other sort of indirectly selected things like recombination, dominance, and ploidy might be susceptible to this thing we're calling sign inversion. I didn't write that part. That's a little over the top for me, but yes.
So, if how is a population able to grow in size if it's sort of passing through a higher mutational pressure, right in the valley? I mean, is that, suppose you start with a population who is growing in size, and then they have to pass through a population size where the deleterious mutations are exerting a great pressure on them.
Mm-hmm.
And even though they're still able to, keep growing in the population size where they get to the beneficial and favored zone.
So what's being plotted in these plots is not growth of a population. It's the relationship between NP fixed, the probability of fixing a mutator, and the population size. So populations at any of these sizes really can grow fine. Ones way down here at a high mutation rate are ultimately going to go extinct because of accumulation of mutations. But so we're not plotting growth here. You ask an interesting question, though, because, and it gets at sort of something I've been talking about and around for all three lectures, which is that we now know there are mechanisms whereby high mutation rates can evolve in populations that don't have any recombination. In the short term, those high mutation rates, well, they must in some sense be favored by indirect selection when they arise and spread through a population. In the longer term, though, they could have quite serious consequences for the population. And Paul mentioned this last night as well. So to me, it's almost as though there's a lag between the effect of beneficial mutations in driving the mutation rate upward and the ultimate effect of deleterious mutations in decreasing fitness. My friend Phil Garish, when we were early in our work on this, said, think of a mutator as a loan shark. It gives you a chunk of money up front. That's the beneficial mutation. But then you have to pay big later. So that's perhaps one colorful way of thinking of it. Okay, what do we have? About 27 minutes left. Was there another question? Yeah, sorry.
Can you just go back to that same slide?
Gladly.
So I was just curious. So there's this threshold which is the boundary selecting, which favors.
Yeah, right. N crit.
Right.
Right.
So is it given a particular species, can you analytically predict what that boundary is?
If you knew the parameters we know for experimental populations of yeast,
Okay.
yes, you could.
Okay. But like I'm just saying,
That's a good question. I mean, I think probably a better way of thinking about that would be, and maybe this is what you're driving at, do we have any reason to suppose that in other populations the parameter space will be so different from the one we studied here that this effect doesn't hold? And I would say no. I can't think of any reason why the parameter space would be so radically different.
it was just
There are some populations in which it's different, but
like it was just motivated by, let's say, if there's a bunch of humans living on an island, is there at a particular point where it falls below a certain level that deleterious mutations will go up greatly?
So that question has really, there are two answers to that question. First of all, humans have recombination. So except insofar as regions of the genome are linked and you might get beneficial mutations and mutators in humans that are in linkage, we don't predict mutator hitchhiking in creatures like humans. But there's a very interesting perspectives paper in genetics, when was it, 3-4 years ago, by Mike Lynch, that caused all sorts of controversy because he suggested that the human mutation rate is sufficiently high that we are, especially with the intervention of modern medicine, individualized genomics, and so forth, accumulating mutations at a non-zero positive rate as a population. And I'm not going to talk about that anymore, but it's a scary paper, and there were responses to it, arguing with it, and so forth. Yeah.
Yeah. There's just a quick question about this equation. So I'm not clear on why you can write this summation only for a mutator and not any other kind of mutation.
Oh, you could, but we're interested in a mutator. We're interested in a mutator, and in particular, a mutator affects its own probability of hitchhiking, right? Because it increases the rate of mutations in its background. In fact, that's what underlies the whole Chow and Cox phenomenon that we looked at in an early slide. Yeah.
So are they in this kind of large N regime?
You bet. Yeah, the things in the Garish paper that predicts the mutation rate, catastrophe or whatever you want to call it, those are large populations. And so
Consistent with that.
and so then the question is, if you started to have a mutational meltdown, would you get down to small enough, that it raises all sorts of interesting questions. Yeah, it's why I'm not agnostic, because that means by definition cannot know, but I'm a little skeptical still of our paper from 2007. There's a ways to go with that idea. Okay. I have some other things I'd like to show you, and we have a little time, so let's do it. So I've written here, at the beginning of my 3 lectures, I said evolution of the genetic system, things like mutation rates, recombination rates. And so, and I showed you a very simple figure, the figure with the red circle and the green square and linkage versus not and so forth. Recombination is clearly expected to inhibit mutator hitchhiking because it erodes linkage disequilibrium or non-random association between mutators and beneficial mutations. And I don't know if was the concept of linkage disequilibrium done in someone's lectures? Okay, well, I wrote some stuff on it up there just to remind you, right? Okay, so there's been relatively little theoretical work on the effect of recombination on genomic mutation rate. It's kind of surprising. I guess that may be because people regard it as a foregone conclusion, but I think there's room for work, because out there in the natural world, mechanisms and rates of recombination vary greatly. Typically in creatures like ourselves, recombination is reciprocal, and it occurs at a fairly high rate per meiosis. But in other creatures, recombination is one way. There's a donor and a recipient and so forth. So there's lots of room for thinking about this. But there's even been less experimental work in this area. And I'm just going to show you 2 projects that are out of my group. One was done by Eugene Rains, who was the driver of the previous work I just showed you, and the other by my current student, Ben Sprung. The first one's published, the other one is, as I say here, in limbo, but promising. So To talk about the published one a little bit, I'm sorry, this is so fuzzy. I realized I didn't have a figure, but I have to tell you a little bit about the yeast life cycle. So yeast, it's the creature that makes bread and beer for us. And it lives out in nature, and it's a creature that has what's called a haplodiplontic life cycle. It alternates between a haploid cell state, and a diploid cell state. And it transitions from the haploid to the diploid cell state by fusion of two haploid spores to form a diploid. And when times get tough, that diploid will go divide out in the environment and have a good time, but in particular under nitrogen starvation, the diploid will undergo what's called sporulation. And that is, it does meiosis, produces the 4 meiotic products that are new haploid spores, and those at some other time will fuse together to form a new diploid. So yeast is a sexually reproducing organism. It doesn't have males and females, it has A's and alphas that fuse together. So we can do, in principle, experiments that involve sex and recombination in the lab with yeast. I have to tell you that it's very time-consuming and laborious, because when you propagate sexually reproducing yeast populations in the lab, they very quickly want to evolve to be asexual. And so you really have to go out of the way to keep them sexual. So that's all that was about. And this is all I'm going to show you from that experiment. This is a paper we published back in 2011. Let me tell you what we did. We did Chow and Cox experiments. Yes? So diploid to haploid transition happens, the diploid's growing out there in the natural environment, and when it encounters tough conditions, in particular nitrogen starvation, nitrogenous compound starvation, the diploid undergoes part of its life cycle where it does meiosis, cases itself in something called an ascus, and produces 4 desiccation-resistant spores, which are the meiotic products, and those spores are haploid. When times are good again, those spores can germinate, and they can grow as haploids, but yeast has this amazing mechanism where, you don't want to hear all this, but basically, if an individual A spore germinates, one of its progeny will switch to an alpha, and they can mate together to form a diploid. Or they can outcross. It's an amazing life cycle. It really is. So what we did here was to do Chow and Cox experiments in sexual and asexual yeast populations, okay? And so what's shown here on the left are mutator dynamics in asexual populations. And let's look at panel B first. So these are diploid asexual populations. So we can propagate the diploids without letting them have sex, right? Celibate diploid populations right here. And it's a Chow and Cox experiment. The mutator goes in at a frequency of around 0.5. It's quite beautiful, nice little lag and then a sweep, and it's what you expect from Chow and Cox, okay? Now look over here. Here are mutator dynamics in sexual populations. Now, what did we do to make them have sex? We had to grow them, then put them through sporulation, isolate the spores, mix the spores at random so that they would mate with each other at random, reconstitute diploids and put them back in the experimental circumstance. And we verified that the spores were mating at random. It was actually a lot of fun. We had markers on the spores and we looked for Hardy-Weinberg frequencies of the mated diploids, and we got them. So we knew that it was at random, or at least we couldn't rule out that it was random. And that was exciting because in a sexual population, the mutator drops out. So we thought, there we go. That's what we expect. If you have recombination, the mutator is not favored. Then we thought we'd better do mutator hitchhiking in haploid populations, not diploids, haploids. And so we started them here at the same frequency, about 50%, and we got a nasty surprise, which is that in haploid populations, the mutators tended to drop out. And this was at large effective population size. What explains that? Well, then we, decided we'd better be better yeast geneticists and see if there's anything known about deleterious mutation rates in yeast. It turns out very high and biased toward very strongly deleterious mutations. This result here really gave us a strong caution that perhaps seeing the mutators drop out here in the sexual populations wasn't a consequence of recombination, but was a consequence of selection in the haploid phase, because the sex experiment had to have a haploid phase. We had to isolate haploids and mix them together. And so we were very honest about this in our paper, and sometimes honesty pays because they published it anyway with our explanations. Okay, here's another one. This is the experiment that's in limbo. This is my student Ben Sprung. And what is this? Well, this is sort of a Chow and Cox type experiment. Here we have mutator frequencies, and this is in E. coli, and this is days out here, so time on the x-axis. We got from our collaborator Katie Cow out at Texas A&M a strain of E. coli that she'd made in which she'd put 3 origins of transfer into the genome. E. coli has I'm going to try not to go into too many details. E. coli has something called an F plasmid that lives in it. Usually it lives as an independent plasmid, but it can integrate into the genome. When it integrates into the genome and an F plus and an F minus cell see each other, a pilus, a structure between them, forms, and DNA gets transferred from the F plus to the F minus. E. coli sex. And then that DNA at some rate will find homologous sequence, and at some rate will recombine in to the recipient cell. And so what we're trying to do here is to engineer up an E. coli that's really sexy, that wants to transfer DNA. And so we got this. This experiment took tons of time, and the results are not, in my view, good enough to publish. But so what we have here is red. The red ones are the things that have the highest transfer rate. The gray ones are the things that have the next highest transfer rate, next highest rate of sex. The black ones are the things that have the lowest rate of transfer. And I won't go into any more of the details, but what you can see is that just by eyeball, there are some populations where the mutator seems to stall out in its climb toward high frequency, and those are, in general, the red populations with the highest rate of sex. So we have done a lot of things. We've gone in and sequenced these guys. We've done all sorts of things. The problem is that the signal isn't very good, and we're now thinking that actually the rate of recombination, even though we've really juiced up this strain to do a lot of transfer, the rate of recombination might not be high enough in this experiment to sort of eliminate mutator hitchhiking. There's one other thing that Ben found, which is, this is, if you look on days 13 and 14, which if you look back here, that's sort of right in here, right about there. He found that the recombination rate is negatively correlated with the mutator frequency on that day, but that's just on that day. So we have a lot more work to do. There really hasn't been a clear sort of piece of work that has sort of started us into a careful experimental, or I would even say theoretical investigation of the relationship between these two really fundamental aspects of genetic systems, recombination and mutation. They affect each other, as it turns out. And I'll say one other thing before I just see if I have time to talk a little bit about the evolution of sex and recombination itself. And that is that most experimental evolution, and here I'm criticizing myself as well, most experimental microbial evolution to date has been, I would say, putting it very provocatively, the exploration of a counterfactual. And the counterfactual is a population with no recombination. Because most populations in nature, even most populations of microbes, have some degree of swapping of genetic material between them. And so it's high time in experimental evolution that we moved toward explorations of recombining populations. The problem is this: there's no single-celled organism on the face of the earth that I know of that is obligately sexual, that you can just plunk into an experimental protocol and expect it to recombine all the time. Yeast, for example, is not obligately sexual. It's perfectly happy to go along without having sex and recombination. If we could find such an organism or make one, then I think there's, lots of scope for interesting work. I think maybe I won't get to talking about the evolutionary advantage of recombination in sex because there are just a few minutes left. Instead, someone asked me a while ago, are there comparative data on mutation rates? There are. I'm at work updating this figure. This is actually from a 2001 or 2000 paper. These are old data. But this is a figure that was put together by one of our co-authors for that paper, Toby Johnson. And let me just sort of take you through this figure a little bit as a way of kind of closing out, and then I'll give you some closing thoughts. So this is a log scale here, and we're plotting two different kinds of mutation rates as known at the time. The total genomic mutation rate, mu sub g, and estimate of the per base pair mutation rate, mu sub bp. And this is genome size in base pairs, and these are a whole bunch of different creatures. And let me tell you what they are. These guys up here with the really high rates, these are RNA viruses. And then when we get here to M13 going down this way, almost in a straight line, are a variety of DNA-based microbes. M13 is a phage, lambda is a phage. These guys, T2, T4 are phages. That's E. coli. There's S. cerevisiae, yeast. There's Neurospora crassa, the bread mold, okay? And then we get to metazoans, C. elegans, Drosophila melanogaster, mouse, human. Okay, so here we're looking at base pair rates. Notice that we metazoans have pretty low per base pair mutation rates. But when we, because of our genome sizes and in humans, because of the number of generations in particular in males between germline replications or transmittals, we actually have quite high genomic mutation rates. In fact, this estimate here is low. In reality, the human, because of new things that have been discovered, the human genomic mutation rate is very high. It's on the order of tens or even a couple hundred mutations per newborn. Not all of those affect fitness, but nonetheless, they're there. There's a curious pattern with mutation rates among microbes that for many years has been called Drake's rule, after the guy who discovered it, Jan Drake. And that is over orders of magnitude of genome size, there's a relatively constant per genome mutation rate in these microbes. The most recent good explanation for this is work on the drift barrier hypothesis of Mike Lynch, who's published some papers on it. I'm not going to go into the details of that. Anyway, I show this figure just to show you that there is variation among populations in mutation rate, and that overall, mutation rates per base pair are quite low in organisms. The kind of pathology that I've been looking at in the lab and talking to you about, it pops up in nature. It's not impossible in nature. I showed you some titles of papers in which mutators were found in natural populations. But in general, it doesn't occur in nature. And what interests me is why doesn't it? Why don't natural populations have higher mutation rates, given the weakness of selection to decrease the mutation rate? If I had to hazard a guess as to why it is, my best guess right now is the presence of recombination in natural populations. Mutation and recombination affect one another. Ironically or paradoxically, it's a population with a high recombination rate that would be most likely to profit from having a high mutation rate, because then that population would generate beneficial mutations at a high rate, but strip by recombination the deleterious mutations away from the beneficial ones, so the beneficials could rise to high frequency without being loaded. But on the other hand, recombination also makes it very difficult to evolve a high mutation rate because the hitchhiking phenomenon is inhibited by a high recombination rate. So I think I'll leave it there. Thanks.
Thanks. It's more than a question, is like confused ideas. So, like in hapodiploids organisms like social insects.
Yes.
So where like the sex ratios are like sort of important for like following like Hamilton arguments, I guess,
right,
about like cooperativity, etc. So is this like the kind of the choice of like these sex ratios or like these being hapodiploid is a kind of evolutionary mechanism in these ideas that you presented, like to cope with.
So could we think of it as an evolvability mechanism or as an evolution of genetic systems kind of question? Is that?
Yeah, that's.
Yeah, I don't know about evolvability, but then I'm generally a skeptic as a sort of starting point about evolvability as adaptation arguments. So yeah, I'm not sure how to address that question. Let me mull it over, and we can maybe catch you later and see if I have anything sensible to say about it. I wouldn't say no. I'm just not sure what I'd say for yes. Yeah.
In sledgehammer selection experiments, you see a frame shift mutation, and it's like kind of persistence in Lenski's experiments as well.
Right.
So in this scenario, you have this clearly fluctuating environments.
Yeah.
And you see this in Lenski's experiment when its environment is almost same.
Yeah, it's constant. It's constant.
Then,
and so how do you reconcile those? I think that the thing that's in common between those experiments is that selection is strong. So you don't, to favor a mutator, you don't need a fluctuating environment. What you need is sufficiently strong selection to propel a mutator to high frequency against the effect of deleterious mutations building up on the mutator background. And so as long as selection is sufficiently, positive selection is sufficiently strong, there's the possibility for hitchhiking. And a sort of nice confirmation of that is that for one of the populations in Lenski's experiment, it evolved a mutator, but many thousands of generations into the experiment, when selection was weak and fitness gain per generation was pretty low, that mutator, a modifier arose, a mut y, which decreased the mutation rate of that population a little bit. And, you know, there's a paper by Wielgoss, I guess is how you pronounce his name, in which they make a very nice argument that reduction of the mutation rate was not likely until sort of the prevailing strength of beneficial mutations coming into the population was small enough that the load reduction could be favored. Yeah. So that's the common thing, is strong selection. That's the common thread.
Related to what you told initially. So over here you're seeing a lot of these organisms near the same mutation rates. And you also talked about same solutions being found to increase your mutation rates. So is it that these solutions, which are commonly found, convergently found in many different experiments, could you find them in natural populations, the sequenced genomes of natural strains which you have?
Yeah, so the question was, if you looked in natural strains, would you find convergent solutions for the evolution of mutation rates? But not the ones that produce high rates that we find in the lab. Instead, what you find, well, first of all, some of the solutions that you see among existing organisms are not convergent. They were descended from a common ancestor, right? So the pathways have, you know, so polymerases, for example, are common to all life. So yeah, there are similar mechanisms, but I don't think, I think a way of looking for that kind of thing might be to ask, if you were to go out in natural populations and find extraordinarily high mutation rates in natural populations, would you see convergence? Yes, you do. In particular, in bacterial populations, if you find mutators in a natural bacterial population, they're very often mismatch repair mutators. Not always, but very often. And I've wondered about this. I think some of it has to do with the target size. That's a multiple locus pathway, and so it's a larger target size. Some of it has to do with theory that suggests that under certain conditions, it's a mutator of intermediate strength that's most likely to hitchhike. A weak one won't get enough beneficial mutations. A very strong one will make too many deleterious mutations. And so there are some similarities.
Yeah, so what you showed with the T locus, I think, right, was there was this expansion of this poly G track. So I'm just curious, like when you look at these mismatch repair enzymes, do you see a selection for synonymous codons which increase like the average length of a repeat? Like for instance, would you swap out a codon which would make it more likely for you to have like a tract of G's or C's or so?
So I'm not exactly sure how to answer that. It is the case that in mismatch repair backgrounds, there is a tendency for an increased instability of repeats in certain mismatch repair backgrounds. So yeah, in that sense, yes. Yeah.
Sir, we know that the mutation rate of the human genome is about 10 to 100 mutations per generation, per meiosis. So there are studies correlating the site of mutations and the site of recombination hotspots in the human genome.
Yeah.
So you see a correlation of more mutations in the hotspots of recombination. So I was wondering how to correlate the relations that you said, more recombination.
That, yeah. Well, I mean, that could be a mechanistic, the correlation could be driven mechanistically. So, you know, one of the things I always scold myself about when I'm thinking about these things is to think that because we have, as humans, have categorized the processes separately, mutation, recombination, gene conversion, pick your favorite thing, that those are in some sense real to the organism, whereas it could be that mechanistically these processes are entangled with one another, and that, for example, recombination is mutagenic. That sort of answer, I think, is a very likely one. I mean, there are other things to say about this, though, which is that there are population genetic factors that will decrease the standing genetic variation in regions of low recombination versus high recombination, background selection being an example of that. And insofar as you are estimating from standing sequence variability in a population, the mutation rate from nucleotide diversity, if you're in a region that has a high recombination rate, perhaps it's no surprise that it has a higher nucleotide diversity than a region with a low recombination rate, which either is subject to background selection or had a recent selective sweep, which purged genetic variation, including neutral variation. So the answer, there are subtleties to the answer there.
Even demographic histories of population have an effect.
Right, exactly right.